Welcome back. So we're going to get right into these different enzymes and toxins that can cause damage to the host. An exoenzyme is an extracellular enzyme that allows pathogens to invade tissue deeper. So you've already invaded the host, but now you're trying to get further in. Maybe you want to get through the cells and into the blood. An example would be hyaluronidase. This breaks up hyaluronic acid, which is found in connective tissues. It is also found helping to bind tissues together. And this allows the pathogen to get into those connective tissues deeper. Examples of connective tissue, blood is a connective tissue, bone is connective tissue, tendons are connective tissue, cartilage is connective tissue. So we have a lot of connective tissue in our body, and if we have pathogens getting into our connective tissues, that's going to cause a lot of problems to our body. Another enzyme that damages connective tissue is collagenase, which digests collagen. Collagen is the most abundant protein in connective tissue. So this is showing dense irregular connective tissue, which is actually found deep to the epidermis. And so if we damage this, it's not only going to damage the connective tissue deep to our epidermis, but it's going to cause our epidermis to also slough off. And that can actually lead to problems like gangrene. Then we have toxins. So toxins are poisons that assist the pathogen in being able to invade the body. So these toxins can be endo or exotoxins. Endotoxins are produced and released by gram-negative bacteria only upon death. And the reason they are released, the reason they're only in gram-negative bacteria is gram-negative bacteria have the lipopolysaccharide that contains the lipid A. And when they die, that lipopolysaccharide breaks, so their cell breaks down, lipopolysaccharide gets into the system, and lipid A is what causes the endotoxin response. So if you have a low level, like you have a few cells that have died, your immune system typically will react and break down those toxins before they can cause severe damage. But if you have high concentrations, so think about severe E. coli infection, GI tract infection, that's going to potentially lead to endotoxic shock. Endotoxic shock causes a high fever, an extremely high fever. The high fever is in response to the lipid A being a pyrenogen, and pyrenogens are substances that cause your hypothalamus to set at a higher temperature. So the more lipid A you have, the higher your temperature is. That extremely high temperature can lead to an extreme increase in inflammation. It can lead to eventually your blood pressure declining. It can lead to multi-organ failure and death of the individual. So it's very, very serious. And so this is just a picture from the chapter 3, which you've already seen, but I'm showing it again. Here you see gram-negative bacteria. They have two plasma membranes. The outer membrane has the lipopolysaccharide associated with it. Here's that lipopolysaccharide pulled out, and you see the lipid A right here. So when the cell dies, that lipid A is released, and that's what causes endotoxic shock. Interesting thing about gram-negative endotoxic shock is that no matter what gram-negative species, you have the exact same response. So endotoxic shock always occurs in the same mechanism. if you have an endotoxin in your system and it is localized, then an inflammatory response can actually clear up the infection relatively fast. It keeps that endotoxin in one region, and then your immune system can respond. But if it's systemic, then it's going to cause a lot of problems. The typical reason for endotoxin shock is due to pathogens dying, typically associated with your immune system, either your innate or your adaptive defenses attacking, or if you're taking antibiotics that kill off the pathogen. These endotoxins are heat stable because they are lipid based, they're not protein. And they are only produced by gram negative. So you cannot get endotoxin shock from a gram positive species. Exotoxins are different. So they differ from endotoxins in multiple ways. So they're proteins. And in general, you don't need a lot of exotoxin to cause damage. You can have very little exotoxin and you can have a very severe response. They are produced mainly by gram negatives, but mainly by gram positive, but certain gram negative bacteria can produce some exotoxins as well. The pathogen releases the exotoxin, the protein, while it's still alive, so it doesn't have to die to release this. And the effects vary depending on the type of exotoxin that was released. Exotoxins are heat labile, which means they can be broken down in high temperatures. So whereas endotoxins are heat stable, these are heat labile. They do not stay functional in high temperatures because they are proteins, and proteins break down or denature in high temperatures, depending on the type of protein. And the effects then depend on the type of exotoxin that is produced. So there are different types of exotoxins. The first one is called an AB exotoxin. These have two parts, which is where they get the A and B. The A is the toxic portion. It is an enzyme, and the B is the binding portion. So you have to bind to a cell, and so that B portion is going to determine which type of cells can be infected. But once bound, then the enzyme can move into the cell and cause structural damage. So common AB toxins: diphtheria toxin, cholera toxin, botulinum toxin, and tetanus toxin are all types of AB toxins. Diphtheria is a cytotoxin, which means it damages cells. And the cells that it damages, so the cells that it attacks actually get damaged and die. Whereas cholera damages, it's an enterotoxin, so it attacks only cells of the GI tract, and it causes those cells to secrete large amounts of fluid, which means you're going to end up releasing a lot of fluid in your feces, causing severe diarrhea or dysentery, and leading to dehydration and death. Both botulinum and tetanus are neurotoxins, so they damage nerve cells, or they cause problems with nerve cells. Botulinum toxin inhibits the release of acetylcholine, causing a flaccid form of paralysis, whereas tetanus inhibits the release of inhibitory stimulations to those excitatory neurons, which is kind of a big mouthful, but basically it stops inhibitory neurons, so your excitatory somatic motor neurons keep stimulating your muscles, and so your muscles continuously contract, forming a spastic paralysis. And so here you see botulinum toxin, and here is tetanus toxin. So this is a somatic motor neuron, and this is a muscle fiber, okay? Normally, acetylcholine is released into the synaptic cleft, and the acetylcholine, which is your excitatory neurotransmitter, binds to the receptors on the muscle fiber in the synaptic cleft. And that stimulates the muscle to contract. What botulinum toxin does is it binds to the motor end plate of the neuron. So these these vesicles containing acetylcholine can't release any of their acetylcholine into the synaptic cleft, and the muscle fiber stays relaxed. Tetanus toxin here, same response. You're constantly releasing the toxin, right? Or you're releasing the acetylcholine, which allows the muscles to be able to function. So again, here that's normal. We have these inhibitory neurons that release inhibitory neurotransmitters. These inhibitory neurotransmitters bind to the motor end plate of the neuron, of the excitatory neuron, and that causes the excitatory neuron to stop releasing acetylcholine. So here's normal, right? And here we have acetylcholine not being released because we have inhibited it by this inhibitory neuron. When tetanus toxin comes in to play, it blocks the inhibitory neurons, and so the excitatory neuron continues to release acetylcholine, causing that spastic paralysis. So both are neurotoxins. Then we have membrane damaging toxins. These are cytotoxins that cause damage to the plasma membrane of cells. They can be hemolysins, which damage or kill, break down red blood cells. They can be phospholipases, which cause the phospholipid membrane to break down. And in all cases, they kill the cells, causing the damage. And so streptolysins from Streptococcus pyogenes causes hemolysis of blood cells. Phospholipase or Clostridium perfringens breaks down the plasma membrane of cells, causing gangrene. Then we have some exotoxins that are called superantigens. So what superantigens do is these toxins cause your innate defenses to go into overdrive. So you basically end up having a cytokine storm. So the toxin gets into your system and your body is flooded with nonspecific cytotoxins or cytokines from helper T cells. So helper T cells typically stimulate your immune system and your nonspecific defenses. But when you are overstimulated, this can lead to an extreme high fever, again, organ failure, low blood pressure. So this is similar. I mean, you have an intense inflammatory response. It's similar to endotoxins. But with superantigens, you don't need a lot of the pathogen to cause this. So toxic shock syndrome is an example of a release of this toxin into your system that can kill you just by a small amount of toxin because it is a superantigen and it causes that cytokine storm. So this is just a table that shows exotoxins and endotoxins, which types of bacteria can produce them, what their composition is, the effect on host, if they are heat stable or heat labile, and then lethal dose. So exotoxins, you don't need a lot to kill. Whereas in endotoxins, you do. You have to have a lot of pathogen to kill you. But just because you need lower dose, that doesn't mean that gram-positive infections are worse. We actually have more mechanisms of controlling gram-positive infections right now than we do gram-negatives. That doesn't mean that we have enough, because a lot of our gram-positives and even gram-negatives are starting to become protected. They're resistant to the different mechanisms we have. But still, I'm just letting you know that don't hope for, if you ever have an infection, an endotoxin, because that can still be really, really hard to control. So if a pathogen, oh, this is a 16-minute video. You know what? I'm going to finish because there's only like 4 more slides. So I'm going to finish. This is going to be a 20-minute video, but your other videos were short. So let's just think of it that way, okay? For a pathogen to be effective, it has to be able to evade our host immune system. So it wants to cause infection, but if it gets killed before it can cause the infection, then it's not very effective. And so we have pathogens that produce capsules to protect themselves from phagocytosis. Some produce enzymes that degrade antibodies. Some produce host enzymes to protect themselves. If they produce host enzymes, then the immune system is not going to recognize the pathogen as effectively. So an example, coagulase produced by an unbroken blood vessel produces a fibrin sheet. That fibrin sheet then can protect Staph aureus. Another mechanism for evading host defenses is antigenic variation. So what this does, it's a mechanism in which pathogens change their surface antigens. So we talked about this when we talked about the protozoan that causes African sleeping sickness, how it can change its surface antigens. So as soon as the immune system starts producing antibodies against it, it will just shift proteins, and then the surface will become a little bit different. And that's what antigen variation does, antigenic variation does. So there are lots of bacteria, I won't say lots, there are bacteria, protozoans, there's even viruses that do this to keep their immune system, to keep the host immune system guessing as to what it is. Once the pathogen has caused the disease, it has done its damage, and now needs to leave. So basically, you've produced lots and lots of cells. Those cells want to leave so they can make their own home. They're going to leave via the same portals that they entered in. So if they entered through the parenteral route, through like a needle or an insect bite, they're probably going to leave through that same area. Like a mosquito might come and bite and take that pathogen out, or a boil might form and the pathogen leaves through the blister. If they get through the nasal cavity or through the respiratory route or the digestive route, they're probably going to leave through those same routes as well. This is the end of your chapter, so I'm going to post these videos, and I'll start on your last chapter in just a few minutes. Okay? And once I find the thing to shut this down, I will turn it off. Okay. What in the world? Huh. Oh my God, what did I just do?